Good morning, everyone. We are Biochem 190 students here to report our review paper on microbial plastic degradation in the Philippines, trends and opportunities in research. As of 2021, the Philippines has been ranked the fifth main contributor to marine plastic pollution globally. On average, the Philippines generates 2,150,000 tons of plastic waste, where 35% of it is leaked to the open environment. Despite this, only a few studies have been dedicated in the country to exploring the ecological aspects of plastics and plastic-degrading microorganisms. With the lack of urgency and physical data, this limits the country's understanding the severity of the problem.
Plastic degradation varies among different types of plastics depending on their form and durability. Oxo-biodegradable plastics are often associated with ocean microplastics, which only partially degrade into fragments after long exposure to sunlight. This results in the formation of barely visible microplastics or nanoplastics in the ocean that can be consumed by animals and easily enter the food chain, which is why its use should be discouraged. These are other types of plastics that end up in oceans. Alternatives, such as bioplastics, have been studied in terms of their limits and waste management process. While there are various disposal methods, they come with detrimental effects, as opposed to a more environmentally friendly process called biodegradation. As stated by 𝗥𝗲𝗴𝗿𝗀𝗀, et al., biodegradation is the process by which microorganisms produce enzymes to break down different materials, such as plastics, into non-toxic compounds. The present review discusses the current trends in research and development related to plastic-degrading microorganisms in the Philippines. Through this review, there will also be an assessment of plastics and plastic additives, their health-related risks, the process of biodegradation, and current innovations in plastic waste management. Lastly, the paper aims to identify opportunities for further R&D of plastic degradation using microorganisms in the Philippines.
Let us now take a look at our results. Over the past decade, the ability of microbes to degrade plastics into simpler organic compounds has been an emerging interest in the Philippines. Here is the summary of some of the most prominent microbial plastic degradation research conducted in the country that were reviewed by our group. As we can see, there are a total of 9 types of plastics that have been investigated so far. Among them are LDPE, HDPE, and polystyrene plastics. As for the microbes used for the degradation studies, the primary focus has been mainly on bacteria and fungi that were locally isolated in various locations such as dump sites, forests, and springs. We will now discuss each of these studies in detail.
Low-density polyethylene is a flexible thermoplastic resistant to the natural process of degradation. The focus lately has been on microorganisms that are able to adapt to extreme conditions such as in high pH or alkaline environments. From a 2018 study, TORRE examined bacterial isolates acquired from a serpentinization-driven alkaline spring with a pH of 11 in Zambales, Philippines. 4 isolates, phylogenetically affiliated with Bacillus 𝗂𝗁𝗄𝗏𝗂, Bacillus 𝗂𝗏𝘀𝗈𝑚𝑢s, Polynoborus fasciculus, and Bacillus sp. exhibited great potential for LDPE biodegradation. This was indicated by weight loss measurements and infrared spectral changes of the LDPE samples before and after the composting process. Other LDPE degrading bacteria have been located in various areas around the country. One of the most promising plastic degrading bacteria, Pseudomonas 𝗬𝗏𝗏𝗏, was isolated from the soil in Payatas dump site. The isolated strains were able to utilize LDPE and PEG as their carbon source. Another strain, Acinetobacter hemolyticus, has gained particular interest from scientists due to its capabilities for aromatic catabolism and hydrocarbon degradation in oil spills. Besides LDPE degradation, several other studies in the Philippines focus on plastics that are much tougher to degrade, such as HDPE. In a 2017 study by Baculi, bacteria was isolated from root nodules of Phaseolus vulgaris. Among the isolates, 4 strains phylogenetically affiliated to Pseudomonas fluorescens and Serratia marcescens showed their capabilities of degrading HDPE film.
Now, moving on to the fungal plastic degradation studies in the Philippines. First, we have the pioneer research conducted by 𝗏𝗏s and Manaligid in 1997. They were able to isolate a potential polyethylene degrading fungi using the baiting method. They identified this as Ascomycete sterile dark mycelia, or ASDM, which they found growing on a sandoplastic bag buried in the forest soil and litter in the lowland secondary forests of Mount Makiling and Laguna. They grew ASDM on a mineral medium containing 0.5% glucose and polyethylene plastic strips as co-carbon sources. Then after 30 days of incubation, they observed a loss of 9 milligrams or 15% of the original mass of the plastic strips. Also, 40 milligrams of dry ASDM was produced. Then, in 2001, the plastic colonization by the same fungi was further investigated by Clutario and 𝗏𝗏s. The two cited that unpublished culture studies have shown that the sterile dark mycelia is actually a fungi called 𝗓𝗨𝗧 sp, which is commonly known as dead man's fingers. To identify the optimum conditions, they grew 𝗓𝗨𝗧 on 4 different media with 4 different sources under different temperatures and pH. The results show that the optimum conditions are at 25 degrees Celsius and pH 5, where 𝗓𝗨𝗧 sp grew more profusely versus non-optimal conditions. This can be seen on the upper SEM image on the left side. Moreover, striations were observed, further confirming that 𝗓𝗨𝗧 has successfully penetrated the polyethylene plastic. Again, 𝗓𝗨𝗧 was applied in a 2019 study by Abesha, Igloso, Tabanlar, and Santiago. But this time, its plastic degrading ability was tested on a polystyrene plastic, and wild-type and mutant strains of the fungi were grown, where after 20 days of incubation, growth and biofilm formation of 𝗓𝗨𝗧 were observed for all strains, as shown here, indicating that 𝗓𝗨𝗧 can also potentially degrade polystyrene. SEM micrographs were also taken after 50 days of incubation, and these images verified that the fungus was able to colonize and physically damage the surface of the polystyrene plastics, as evidenced by 4 things. One is the mycelial mats, as shown here. Second is surface cracks, as you can see in this image. Third, crevices that were observed here. And lastly, holes that can be seen here. Aside from 𝗓𝗨𝗧, other potential fungal plastic degraders have also been studied by Urzo, 𝗏𝗏s, and 𝗏𝗨𝗀𝗀 in 2017. They tested 18 different lignin and cellulose-degrading fungi on a medium containing polyester polyurethane plastic. From the 18 fungi screened, the following 4 fungus isolates showed potential for degrading polyester polyurethane, where 𝗣𝗏𝗀𝗏𝗃 appeared to be the slowest in utilizing the said plastic. It is also notable that this study was the first to report the formation of crystal-like structures in the culture containing the degraded polyester polyurethane, which may be part of a novel mechanism of fungal plastic degradation.
Microbial degradation is influenced by several factors, with the major factor being plastic composition. Non-biodegradable plastics have polymers that only become more susceptible to microbial degradation under extreme conditions. Biodegradable plastics, on the other hand, are typically made from renewable raw materials, such as starch or cellulose, that are easier to degrade. Soil burial tests were also undertaken for 3 types of biodegradable plastics to investigate the degrading abilities of the natural soil microflora. With the non-biodegradable plastic as a control, researchers compared the biodegradable samples from SM Supermarket, 7-Eleven, and Robinson Supermarket. Using 2 bacterial species, Cellulomonas flavigena and Arthrobacter luteus, degradation was measured through weight loss. Compared to other plastics, BP3 was more readily degradable. This outcome was attributed to a higher starch content in Robinson Supermarket plastics. Biodegradable plastics can be more toxic than pure plastics. Additives are added to the product to make it more durable. According to Lumio 2021, these additives typically comprise 10%
to 70% weight per weight of the plastic and are highly toxic even at a nanomolar concentration.
The ability of microbes to degrade non-biodegradable versus oxo-biodegradable plastics was also looked into by Gutierrez and her team. They tested the degrading ability of 𝗤𝗏𝗀𝗁𝗃, a white rot fungus, where they found that it degrades the non-biodegradable LDPE plastic at a higher rate than OBP. These findings further support the dangers of considering OBP as biodegradable. Nonetheless, SEM reveals that the fungus was able to physically damage both LDPE and OBP surfaces. In addition, the group further studied the effect of the environment on the degradation rate of the fungus, using Sabarod's dextrose agar medium for the lab conditions compared to soil. And their findings demonstrate that the fungus exhibits greater plastic degrading ability in the laboratory medium versus that in soil, as we can see on the steeper red lines for the medium versus the blue lines for the soil in both graphs.
Now let's go into gut microbes. So studies have shown that some insect species are able to consume plastic and use it as their primary carbon and energy source. So for example, Bombelli and his team were able to show that the larvae of wax moth Galeria melanella biodegrade LDPE into ethylene glycol byproducts. And after 3 years, a study was able to prove that this biodegradation is largely caused by the intestinal microbes, mostly from the Acinetobacter genus. A similar research was also performed with red floor beetles, which are serious agricultural pests here in the Philippines. In this study by Bacterial and Almeerah, the beetles were fed with expanded polystyrene, and results showed a decrease in the mass of the polystyrene after incubation. However, currently no further research were made to determine whether this biodegradation is caused by its gut microbes or not. After learning about many of the current and local studies done on microbial plastic degradation, let's now go into the opportunities for further research. So here's a graph of the 2019 data on the plastic waste generation here in the Philippines. And if you can see, the two plastic types that constitute most of the waste are polypropylene and polyethylene terephthalate. However, although PP and PET constitute most of the plastic waste generated in the country, most microbial plastic degradation studies here focus on LDPE, HDPE, polyurethane, and polystyrene. This suggests that further degradation researches on these two plastic types are very crucial. So one research topic for PET degradation that is a current trend in the global scale will be about PET degrading enzymes, specifically the two enzymes phtase and metase, which were isolated from the bacteria Ideonella sakaiensis. This was a very huge discovery, as with only these two enzymes, PET can be degraded to its monomers, which is ethylene glycol and terephthalic acid. However, the capability of this bacteria is limited by many factors such as its habitat. With this, recent research delves into allowing the bacteria to survive in soil and water, where most plastic wastes are found. There's just so much potential with this enzyme, and further research regarding this will be valuable. Research on plastic degrading microbes for marine and freshwater areas would also be incredibly important, since most of the plastic wastes are not only found in the country's dump sites and landfills, but also in these areas. Another important topic to further study on will be the biodegradability of the alternatives of non-biodegradable plastics. For example, for a period of time, oxo-degradable plastics were advertised as more eco-friendly. However, it was later known that it causes even more harm than non-biodegradable plastics, since even though the additives added in the plastics help in degrading it into microplastics, these microplastics will still persist in nature and cause serious health and environmental problems. Furthermore, the plastic additives themselves are also important to research more about, since when plastic waste gets degraded, these additives gets released also, and the accumulation of this could also have serious impacts. To conclude, numerous researches have been conducted in the Philippines regarding microbial plastic degradation. especially in regards to isolating plastic-degrading bacteria and fungi from dump sites, sediments, alkaline spring waters, root nodules, and soil. Some further studies were also able to determine the optimal conditions, effective supplements, and the mechanisms used for these microbes. Moreover, gut microbes of some insects were also determined to have plastic-degrading capabilities. Some research also focused on the microbial biodegradation of different types of plastics. Lastly, with the current research and innovation of microbial plastic degradation in the Philippines, the paper identifies 4 opportunities for further studies, which are: first, microbial degradation of polypropylene and polyethylene terephthalate; second, further research on phtase and methase; third, microbial plastic degradation for marine waters; and lastly, further studies on the biodegradability of non-biodegradable plastic alternatives and plastic additives. So here are the references. Thank you for listening.